Hello friends, welcome back to my channel, and today in this video we are going to study about overview of receptors and their activation. We learned about this figure in the last video that whenever any ligand bind with the receptor, it brings some conformational change, and that's how the intracellular events will start. Today we will see about, or we will learn about, this intracellular events. Now, what do we mean by signal transduction pathway? Signal transduction pathway means as soon as any ligand binds with the receptor present on the surface of the cell, a cascade of signaling reaction will start, which ultimately go and bind to the gene for its expression. And the expression will depend upon the ligand that bound to the receptor, because the expression is the response against that ligand binding. Now, in the signal transduction pathway, some components are known as upstream components, some components are known as downstream component. So, the components that are near with this receptor molecule, this is a receptor molecule. So, the components that are towards the receptor molecule or closest to the receptor molecule, we call them as a upstream. And the components that are closest towards the gene side or the response side, we call them as a downstream. So as soon as any ligand bind with the receptor, a series of a cascade of reactions will start, which ultimately help in the expression of some genes. Now the overview of lymphocyte signaling. So what we can see here, as soon as any ligand bind with the receptor can be accommodated with any co-receptors and the receptor associated molecules. So what will happen? Whenever any ligand bind with the receptor, some conformational change will occur and phosphorylation will occur. Receptor associated tyrosine kinase. Kinase means that can add phosphorus. So with the help of addition of phosphorus, the further molecules are get activated. Clear? So this receptor associated molecules, they will get phosphorylated, and because of which the adapter molecule will come and bind to that. Adapter means they function as a docking site for the addition of various other signaling molecules. So this adapter will get phosphorylated, which in turn will bind several other molecules, as you can see here. Now PI3 kinase, what it do? Kinase, that means addition of phosphorus. So it will add phosphorus to PIP2 to convert it into PIP3. Now the adapter will help to activate and bind PLC gamma, that is phospholipase C gamma. This phospholipase C gamma will come and bound with PIP3, which is present on the surface. So until this PIP3 is formed, maybe this PCLC gamma is present somewhere inside the cytoplasm. But as soon as this PIP3 is formed, the adapters become activated. This PLCY molecule will get pulled towards the membrane and it will bound with PIP3. Once it is bound with this PIP3, now what it can do? It can dissociate PIP2 into DAG and IP3. Full forms are written here. IP3 is inositol triphosphate, DAG is diacylglycerol. So PLC gamma is an enzyme. The substrate for this enzyme is PIP2. Where this PIP2 is present? On the membrane. So this enzyme should come and bind on the membrane, then only it can bind with the substrate and perform its function. So to bind with the membrane, it needs PIP3. So first of all, PI3 kinase will convert PIP2 into PIP3 so that now this PCLC gamma will pull towards the membrane and remain attached on the membrane for its function. So what it will do? It will break PIP2 into DAG and IP3. What IP3 will do? IP3 will go up to endoplasmic reticulum, because the endoplasmic reticulum surface contains receptor for this IP3. As soon as this IP3 will bind with the endoplasmic reticulum, this endoplasmic reticulum will release calcium. This calcium will then activate calmodulin calcineurin complex. Once this complex get activated, what it will do? This NF80, which is phosphorylated, which is present already in the phosphorylated form, it will remove this phosphorus from this NF80, and that's how the NF80 becomes activated, and it will cross the nuclear membrane and enter inside the nucleus for the activation of the gene. This is one pathway. Second pathway, because here we learned DAG is formed. So now from DAG, the enzyme PKC, again a phosphokinase, it will phosphorylate this complex, NF kappa beta and I kappa beta, because this NF kappa beta is a nuclear factor, so it can enter inside the nucleus, but it remain inactivated due to this kappa beta. So what the PKC will do? PKC will phosphorylate the kappa beta, and that's how it will remove the kappa beta and proceed for degradation. So now NF kappa beta is free, it can cross the nuclear barrier, enter inside the nucleus, and it can activate the gene. This is one more pathway. Now if you see the third pathway, the DAG can also activate the RAS and then the MAP, RAS MAP kinase pathway. This will activate AP1, and that's how it can activate a gene. So for the gene activation, as you can see, whenever any ligand bind to the receptor, there are multiple signal transduction pathway that can function for the activation of the gene. Sometimes any particular gene activation may require only one transcription factor for its activation. So maybe among all the three pathways, any one pathway can activate this particular gene. But for some gene activation, it may require multiple transcription factor. One factor from this NF80, one AP1, another NF kappa beta. So all the three will bind, then only this gene will express. So in that case, all the three signaling pathways will be required. So whenever any ligand bind to the receptor, all the pathways can come into play, or any one pathway also can come into play. That depends upon the requirement of the gene which needed to be expressed. So this is an overview of lymphocyte signaling. So whenever any ligand bind, the first step will be binding of the ligand. The second step will be the activation of the receptor, either by conformational change or by phosphorylation. This will activate any adapter molecule, and this adapter will bring many signaling molecules and will activate. These are the common. And then it can proceed either from NF80, RAS MAP kinase pathway, nuclear factor kappa beta, any of this pathway or all. Now this diagram is showing the activation via conformational change. As you can see, these dimer receptors are present far from each other, but as soon as any ligands come and bind, it helps in the dimerization of this receptor and the cross phosphorylation. That means this segment is phosphorylating this segment, and this segment is phosphorylating this segment. So as soon as the ligand bind, there occur a conformational change. The receptors that are present far now will come close together for the binding of that single ligand, and the cross phosphorylation will activate the receptor. And from with this receptor, then can any adapter will bind, this adapter will bind multiple signaling molecule, and that's how the process will proceed further. Now here one thing you should remember, that if you see, the receptor is quite long in the cytoplasmic end. This is the cytosol. So the cytoplasmic domain of the receptor is quite long for the signaling to proceed further. Now one more activation we can see, that is via receptor associated molecule. Now this is a B cell. What is the receptor for B cell? BCR. And BCR is nothing but any antibody molecule, as you can see here, IgM. So now you can see here the BCR does not have the cytoplasmic domain. So definitely for the signal transduction pathway, it will need the association of other receptor part that will together form an activated receptor for the signaling molecule, because the IgM, the antibody itself, does not have the cytoplasmic domain. So in case of B cell, it contains Ig alpha and beta. Ig alpha and beta, this is CD19, which is associated with CD21. This CD21 has the part of complement, that is C3d. This CD3 can also bind with the antigen to which the antibody is bound. And it also contains some CD81. This is for B cell. If you see the T cell, similarly like B cell, it has TCR, T cell receptor. Now for T cell receptor also, as you can see, the cytoplasmic domain is very short. So it will also require some extra receptor or co-receptor molecules. So this is known as CD3 in case of T cell. Here it is CD21, which bound with C3d, which bound with antigen. Here it contains, T cell contains either CD4 or if it is TC cell, it will contain CD8 which interacts with the MHC. What is different in B cell and T cell is this CD81 in B cell and this CD28 in T cell. So here it is written CD alpha beta or Ig alpha beta in B cell and CD3 in T cell is equivalent. IgM in B cell, TCR in B cell equivalent. CD19 and 21 and CD4 and 8 in T cell equivalent. The difference is B cell contains CD81, T cell contains CD28. So once any ligand bind with the receptor, that is the immune cell receptors, either B cell or the T cell, because the BCR and TCR, they have very small cytoplasmic domain. So this main receptor should be associated with other receptor part having the long cytoplasmic domain that can help to transmit the signal in the cell. For B cell, we have CD19 and CD21. For T cell, we have CD4 or CD8. Clear? So that's how whenever any ligand bind to the receptor, the signal transduction process starts. So that's all for this video. If you found the video informative, please do like, share and subscribe the channel and hit the bell icon for the next video notification. Till then, stay safe, stay happy.